right, so let's add myself in. Welcome everyone. It's nice to be streaming again. It's been a long time, around 200 days I think since I last streamed. So it's good to be back. It's been super busy at work. So it's just been difficult to find time to make anything and set up streams and stuff, but we're back. I can see myself now as well. Perfect. So yeah, it's been busy, but it's been really good to kind of prepare this. So today we're gonna discuss enrichment analysis, also called overrepresentation analysis, pathway aura, gene ontology, overrepresentation. So we're just gonna go slow. We start off, of course, like always, with a little bit of theory, and then we'll start coding. The code is down in the description already. So if you want to follow along, just look at the code and copy paste it in together with me, and we'll see what happens. So that's about it for today. If there's no further questions, then like they say, let's just dive into it. So for today, this is the overview that I made for you guys. So first we'll talk a little bit about enrichment analysis, what it is, what you can use it for. And then I want to talk about today's data set, because of course to do enrichment analysis, you need a data set. So I thought it would be good to kind of not have like a contrived example, but to use some real data. So there's a paper that was published, I think 15 years ago. It's a very old data set. So I selected the data set because it's relatively small. So it runs really fast and you can iterate on it. But it's a nice data set. So we'll be talking about that. There are some packages that you're going to use. So it's not entirely from scratch, but it's going to be very low level. And then I want to talk about the files, how to get them automatically from Gene Expression Omnibus, so that you don't have to go to the website, click on download like 5 or 6 or 20 times, depending on how many samples there are. And then we'll very quickly run through how to do differential gene expression, so how to find differentially expressed genes between two conditions. Make a little volcano plot, just because I love making volcano plots. And gene ontology then. So because I thought about using KEGG pathways or using the Reactome database, but when I downloaded the dataset, the nice thing is it comes with gene ontology for every probe. So I thought, why not just use the gene ontology annotation that's there? So in gene ontology, you have to define a foreground set and a background set. We'll get into that because I made some pictures so that you guys can kind of see what's happening. And then we will do the overrepresentation test. So the overrepresentation test is going to be a hypergeometric test. So in R, there's the phyper data set, or the phyper function to do that. And then we'll spend like 2 slides formatting the output so that you get it really nice, like a normal tool will give you. Of course, there's a lot of like online tools as well. You can use tools like David, or you can use tools like WebGestalt to do these kinds of things. Sorry to ask an irrelevant question. May I know, are the videos of R programming enough to perform complete data analysis of medical data? Yes. It gives you a basics. So if you follow the R course, so that's also what this presentation or this stream is about. It's kind of a follow-up. So it's just using tools that you already have acquired during the R programming lectures to kind of build on top of that. So this is kind of more advanced. It kind of assumes that you have done most of the R lectures. And don't worry, like feel free to ask questions, interrupt me anytime. Just throw it in chat. It's always fun to have feedback from you guys. So if you have a question, no matter how irrelevant you think it might be, just let me know and I'll answer it for you. All right, so that's the overview from today. I'm hoping that we'll be able to do it in like 50 minutes. We might take a little bit more time. but it's 36 slides, and we're only on slide 3, and I've already been streaming now for 6 minutes, so that's 2 minutes per slide, so it's going to be probably slightly over an hour. But you know me, I love talking, so that's one of the things that's going to happen. So we'll get off track, we'll get lost in the woods, but that's all fine. That's all fine. I'll be here until the end, so just stick with me. All right, so next slide. Enrichment analysis, right? I already told you guys it's often called overrepresentation analysis. Overrepresentation analysis is the same as enrichment analysis. It's just two different words for the same thing. So the basic question that it tries to answer is an annotation enriched in my set? Right, so there's two concepts there that we have to go into. So it's a concept of annotation and it's the concept of set. So a set can be anything. For example, a certain number of genes that you found to be differentially expressed. It can be a certain region of the genome where a certain amount of genes are located. But it can also be things like proteins, right? It could be the protein abundance of a certain set or differentially abundant proteins. It can also be metabolites. So I recently implemented this whole pathway overrepresentation for a metabolite data set. So also there you can use it, which is really, really fun. So the annotation is more difficult to explain. Right? A set is relatively easy. It's just a group of genes which are differentially expressed relative to another set, another group of genes which is not differentially expressed. But an annotation is difficult. So annotations are things that humans annotated to genes, right? So you can imagine that if we have a gene in the genome, then this gene is expressed at a certain point, right? So the protein is found to be in the nucleus, or the protein is found to be in the mitochondria, or it's found to be inside of the endoplasmatic reticulum, or it's inside of the cell wall, right? So that is an annotation, right? So if you would go through all of the genes in the genome, and for every gene you would say the location where this gene is expressed is the nucleus, or it's the mitochondria, right? So that is then the annotation of the genes. So there's different types of annotations. So for example, cellular localization, where a gene is found, is one of the different types of annotations that there are. But you have many, many different types of annotations. For example, if you think about pathways, being part of a certain pathway is also a certain annotation. Right, so what an overrepresentation test does is it takes your set, it looks at the set, and then it tries to figure out if any of these annotations is more commonly found in your set relative to a background set, right? So you can think about like a jar, like above, where there's all kinds of different candy in there. So now the question is any one type of candy more abundant in my jar relative to the bag that I filled the jar from, right? So that's kind of the whole idea about enrichment or overrepresentation analysis. So the dataset that we'll be using today comes from this paper, Dissection of DNA Damage Induced Transcriptional Networks. So it's a very complex paper, and today we'll be only focusing on the control samples. So we won't be dealing with the siRNA knockdowns and that kind of things. We'll just take a very basic 3 versus 3, and we'll just start looking at the controls. Of course, feel free that after we're done to take the code, adapt it, and redo the whole analysis using all 30 samples that they have, because then you can just redo what they did in the paper. So the paper also has a gene ontology analysis. So after you're done, you can benchmark your code and compare it to what they did in the paper. So that I always think that's really fun that this paper, which was published in 2005, so it's like 19 years ago, that you can still redo this. So that's awesome. I love it. So the data set is a microarray data set, which means that the expression of around 8,500 genes have been measured using this Affymetrix human focus gene chip. And it's a human chip, so the data that was on there is not humans, but it is HEC293 cells. So HEC293 cells are a very specific cell line made from human embryonic kidney cells. So it's an immortalized cell line which you can just grow and grow and grow. So you can reproduce your analysis like 10 years down the line by getting new HEC cells and redoing the experiment. So we're looking at the control samples. So 3 control samples are untreated HEC cells, and then we have 3 control samples which are treated. And I'm going to mess up this word. I tried to test it a couple of times and pronounce it, but they have been treated by something which is called neocarzinostatin. So neocarzinostatin is a cytotoxic substance which is used in cancer treatment. So the authors are very interested in what happens to human embryonic kidney cells, so a type of cancer cells, when you treat them with this compound. This compound comes originally from bacteria. We need a video on genome-wide analysis of different plants. Yeah, we might do more GWAS. I was thinking about doing a QTL mapping, not GWAS. I'm not a GWAS expert, but I'm a plant expert. So. Anyway, so this is our data set that we'll be using. So the link to the paper is also down in the description. So you can find the paper there and show some love to the authors of the paper, because I do think it's a pretty good paper. All right, so one thing is that the data is publicly available. That's one of the requirements. So it is a publicly available data set. Which is available on GEO. So GEO is the Gene Expression Omnibus. It's a big database from NCBI where you can find all kinds of gene expression data. So like I said, it contains 30 samples in total. Today we'll only be looking at the uninfected control samples. So we're not even going to take the control samples which have been infected with an empty vector. We're just going to take HEC cells which are grown, and then HEC cells which are grown and then have been treated with this neocarzinostatin. So that's the data set for today. I put the link here. Let's very quickly look at the data because I think it's important that people look at the website a couple of times to see how you can find this data yourself. So this is how the GEO website looks. So I'm already looking at the series. So this is 1676, that's the dataset identifier. Again, it gives you a title, it gives you the organism on which it's done. It's expression profiling by array. And then if we scroll down, you can see the authors of the paper, and you can see the citation that you can use to use when you cite or when you use their data. So you can see that it's done on one platform. So the platform is the Affymetrix Human HG Focus Target Array, like I told you guys. And if you would click here, you would go to the annotation of the array. So every array has different probes. Every probe targets a certain gene, and all of these kinds of information can be found here. And then here we have the samples. So if we click more, then we can see that not only do we have the uninfected control samples, but we have the LAC-Z control. So those are the samples in which they added a plasmid, an empty plasmid, not expressing anything. And then we have the different siRNA samples where they knocked out certain genes, for example, P53, which is a well-known kind of clock gene. So it's a gene which is there to regulate cell cycle. All right, so that's the data set that we are going to look at today. So before we start doing the analysis in R, we need to have some R packages installed. So installing packages is done via BioConductor. On the next slide, we'll see the commands to do this. But we're going to use the affy package because we are using an Affymetrix array. So this allows us to automatically load in the cell files from our hard drive after we downloaded them. So Affymetrix is really good in providing annotation and analysis tools for their data set. So it's very easy to work with Affymetrics microarrays. There's nothing that you really have to do. Compared to doing stuff like RNA sequencing, it's much more involved. This is, since it's a relatively older technique, this is a very, very, very streamlined analysis. So we're going to use affy to load in the data, and then we're going to use limma, which is linear models for microarray data. Again, a very, very well-established package. We don't have to do much ourselves. We can just call one or two functions from the limma package to do the whole thing, which is like the pre-processing of the microarray data, as well as the statistical analysis. So we're not going to focus on that during this lecture, because I want to focus on doing the gene ontology, doing the overrepresentation analysis. And we need to have preprocessed core installed. So preprocessed core together with limma, these two packages work together. But when I was testing it under Linux, I found a little bug. So if you are working under Linux, there's going to be an additional command to first install preprocessed core and then install the affy and the limma packages. So just as a workaround, I put it in one of the slides that is going to come up. All right, so we're going to install the packages. I already installed them in my R, so in this case, I don't have to do anything. I can show you guys what the command will do. It will just tell me that the packages are up to date. So I used colors when we do this, right? So when we look at code, I always try to use colors. So the blue color is for functions. The orangey color is for strings. So those are kind of character constants. There's a different color like that will come up, but the purple color here, so the darker purple color, that's for numbers and constants. So those are things that are kind of constantly defined, so we sometimes have to talk about them. But first thing that we want to do is we want to install BioConductor, the bio manager, so that we can download these packages from BioConductor. So this is the code that we can use. So if it is not installed, then we need to install this package. And then we use the BioManager install function to then install these three packages all together. Of course, you can just call the install function 3 times, which would work as well. So let's go to R and let's do that. So let's open up my R window, and I'm just going to say, so I have BioManager installed, so I'm not going to do the first one. I'm just going to install affy and limma, which should install preprocessed core as well. And since I'm under Windows at the moment, I don't have any issues with having to install preprocessed core separately. So I can just press the key, and then it will start downloading. And it tells me that it wants to update all of my packages. So I'm going to say no, we don't have time to update it. But it gives me a warning, and the warning says that the packages were not installed when the version is the same or greater than current. Because of course I was preparing this, so I installed them yesterday. All right, so after we have that, the thing is to make sure that we are knowing where we are going to save our files, right? So I'm going to set my working directory to a folder on my hard drive. So since I'm on Windows, it's C// for the C drive. It's going to be in users, then e-documents, and then the folder that I created on my hard drive. So you just go to your desktop, you right click, and then you just create a new folder. And the folder, in my case, I called it GORA for Gene Ontology Overrepresentation Analysis. And then we're just going to load the different packages. So we're going to load the affy library, the limma library, and the preprocessed core library. So let me switch to R again. And let's load in those libraries. So I'm first going to set my working directory to make sure that I'm there. And then I'm just going to load the different libraries. They will throw a lot of feedback. So they will say that stuff is overridden. But that's just the way it is, because these packages come with all kinds of different functions. So we just have to accept the fact that some of the default functions in R are going to be overwritten by the functions from these packages. All right, so everything seems to be loading all right for me. So in this case, if you have any issues with how to load the packages, it should be relatively straightforward. But in the end, if you have any questions, then let me know in chat now so I can directly look at the issues that you have. Or of course, leave a comment underneath the video later on. So loading the libraries, now we're all set to go. So that's kind of what we have to do. Just install these 3 packages and then make sure that they are active in R. Of course, we only have to install them once. We don't have to every time do the install code. So we just do this once, packages are installed, and then only when we install a new version of R or go to a different computer do we need to do this again. But from now on, we can just start every time with the set working directory and then the 3 library calls. So like I told you guys, when I was developing the code, I was using my Linux machine at work. There's a little bug in Linux, and it leads to an issue with pthreads because the preprocessed core package tries to use all of the cores on the machine that are available. And sometimes you get an error 22. The problem there is that you have to then say disable threading. All right, question in chat. We're interested in knowing the impact of frailty index on the survival. Can you please help me with it? Yeah, sure, Matt. If you want my help, just send me an email and we can look at that separately outside of the stream. No worries. So preprocessed core, like I said, little bug under Linux. So if you want to install it under Linux, make sure that you use this command. You have to give an additional argument saying that the threading needs to be disabled. Of course, if you're running the code and you don't get any issues, then of course it's perfectly fine to use it with threading. But on my machine, so I'm using an Ubuntu 2004, I think. So the Ubuntu version that I was using was having this threading issue. So you just have to install the package then and disable the multi-threading support. It will make it run a little bit slower, but it's better to run a little bit slower than to not run at all. And of course, you have to add force equals true, update equals true, and type is source because you need to force reinstall the package over the old version that you have installed. So initially under Linux, just try to do this. If everything works well, then you're not affected by the bug. If you are affected by the bug and you get like a pthread error at some point, generally it's error 22, but it could be a different one. Then just use this command, close down R, restart R, and it should fix the threading bug that is in the pthread library. All right, so those are the packages. So now we need to kind of hold our data, right? Because we are looking at Gene Ontology, we're looking at Geo, right? Let's go back to Firefox. So when we look at Geo, we have all of these GSM names for our different samples, right? So we have 6 samples because we're only using the unaffected control. So I just need to use these names to download the data from for them. So what I'm going to do is I'm going to create a very, very tiny matrix. And this matrix will have the sample names as the row names, and then it will say the type. And in this case, all of them are going to be control samples. And then I'm going to add another column to it, which is the time. So it's 0 hours. So this is pre-treatment, and this is after treatment for 4 hours. So this is just a little matrix, and this little matrix is going to be used for 2 steps. It's going to be used to download the data, but it's also going to be used when we do the differential gene expression. Because when we do the differential gene expression, we need to build a statistical model where we have to compare the 4-hour samples to the 0-hour samples. So compare the treated samples versus the non-treated samples. So I always use a data frame for this, although it's a little matrix. The data frame is just nicer because it allows you to have different types in the same kind of data frame. So the way that we do it is we do a C-bind. So I'm doing a column bind of these 2 columns. So I'm just going to create a type column by using the repeat function. So I'm going to repeat the word control 6 times. That's going to be my first column. And then the time column is going to be very similar. So I combine a repeat 3 times of 0 hours with a repeat of 4 hours. I'm going to repeat that 3 times as well. Right, so then I combine these 2 little vectors together using the column bind, and then I get a little matrix, and after that I set the row names of this little matrix so that we have a well annotated little data frame. Is there a way to combine the metadata of 2 different data sets? Yeah, of course, you can always make it yourself, or you can just row bind to it, right? If I would have 2 of these matrices, I could just row bind them together to get one on top, the other one below. So let me show you, because we're here anyway. So let's open up R. So let's make this little sample matrix. So I have the code ready. So if I type samples, then you can see that it looks like this. If I would have 2 of these, I can just say row bind samples, and then I'm going to do samples with it. So when I do this, now you see that I have the same matrix twice. So I now have, instead of having 6 samples, I now have 12 samples. So this is how you would kind of extend it, right? If you would want to do all 30 samples, then you would create one of these little for control. You would create one for the samples which have been treated by the empty vector, and you would create something which has been treated by, for example, P53 knockout. But you will just want to create one very large matrix. So generally, the easiest is to kind of keep it simple. First do 6 samples, then add another condition, and then add another condition. But start small and then just build up on top of that. So the row bind allows you to combine 2 matrices by their rows. So that's how you can kind of extend it if you have multiple matrices. So the one thing that you see that happens now, because samples already has the same row names, it will put a 1 behind the row names. So you have to make sure that if you are combining 2 matrices, that they have unique row names. Otherwise, the row names will try to be smart and say, well, no, this is the second one that I see, so I'm going to add a 1 to it. If we would do it again, then you will see that the later samples will now get a 2 added to them. So for a matrix, it's very, very important to have unique row names because R uses the row names to select stuff from it. So you're not allowed to have row names which are duplicates. All right, let's go back. So we now have our samples defined. So this is the first step. These numbers, like I showed you guys, they just come from the website. And we're going to use these to download the data sets from online. So that in case we have 30 samples, we don't have to click download 30 times, put them in the folder, and deal with that ourselves. No, we can have R deal with that for us. So how do we do this? Well, we have to create a little for loop, right? Because we have to do something for each of the samples. We have 6 samples, so we could just say for each of these. So that's what I'm doing here. So if you want to download from Geo, you need to know the download address. So the download address is https://www.ncbi.nlm.nih.gov/geo/download. And then I'm going to make a little for loop. So I'm going to say for each ID that you find in the row names of the sample matrix, I want to do this, right? So it's going to do this as many times as there are row names of the matrix. So in our case, we have 6 row names, so it will do this 6 times. And every time, ID will contain the name of the row. So it will contain GSM 28627. And then the next time it will contain GSM 28628, right? So it will just go through the rows of the matrix, take the row name and put them in the ID variable. So then we built the download URL or URI, Unified Resource Locator. So we built the URI. So what do we do? Well, we are just going to extend this GeoString. So we're going to take the GeoString and then we're going to add to it $ACC for accession is equal to the ID. I'm going to say format is file because I want to download a file. And I'm going to say the file that I want to download is the ID. And then you have this additional section at the end. So since URLs cannot contain any dots, this is the dot. So the %2e, that is a dot in HTML speak. Well, not so much HTML speak, but in URL speak, right? Because a URL can only contain dots within the www.something.com, but after that, it's not allowed to have dots anymore. So we just have to use the %2e, which gets translated into a dot. So we'll download cell files and we'll download the GZ, so we'll download the compressed version of the cell files. So those are much, much smaller. So in this case, I think every file is around 1.8, 1.6 megabytes. So that's not too bad. If you have bigger microarrays, like tiling arrays, you could end up downloading files which are like 200 MBs. But that's why I took a slightly older data set to just show you the concept, and it runs really, really fast. So if we are going to execute this, the downloads in itself are just going to take like 10 seconds to download on a normal kind of broadband internet. I also need to tell the download file function that we want to use to download where I want to save them to. So I'm going to say I'm going to make an FP for file pointer, which is going to be just the ID and then .cell.gz. So I'm using the paste0 function so that it doesn't include any spaces between the different words, because this needs to be a single long URL, and this needs to be kind of the name of the file, and I don't want any spaces in there. So the files that I'm going to save are just going to be gsm28627.cell.gz. And then I'm going to check if the file does not exist already, because if I'm executing the code multiple times, I don't want it to download the file when I've already downloaded it, right? You want to be nice to the server. You want to be nice to the people from NCBI. You only want to download a file once. You don't want to download the same file 30 times if you execute the script 30 times. So be a little bit considerate to the people running this infrastructure, because they have to pay for their server, they have to pay their internet provider. So every time that you download something, they are paying for you, which is really nice of them. So I'm going to use the download file. So I'm going to say if not file exists file pointer. So if this file is not found on my hard drive, then download the file. The file can be found at this URL, and I want to save it into this name, so the FP. And then I'm going to sleep for 5 seconds, because otherwise, if you would download the file, you don't want to continuously hit the external server with requests. So if you download a file, wait 5 to 10 seconds before trying the next file, just so that you don't get blocked. Right? It's always good to make sure that you have all of your timeouts correct. So I think for NCBI, they say wait 3 seconds. In between requests, I'm always waiting 5 to 10 seconds, right? In the end, in the grand scheme of things, it doesn't matter too much that you have to wait a little bit. All right, so let's move back to R and make sure that we download this. So of course the code, you can just get the code from online, so there's no typing involved this time, which is really nice. Unless you guys ask me questions, of course, then I can extend on it. But just let's start downloading the file. So it will just say downloaded the file. So this first file is 1.3 megabytes, and then it downloads the second one. So it tells you the whole URL, which it tries to download, and you get a little pop-up in R where you see a progress bar while it is downloading, which is really nice. But you guys can't see that since I'm only capturing the main R window and not the download itself. All right, so that's 5 files done. So then the last one. So there we go. And 5 seconds sleeping time. And then we should be back in R. So now if I do a dir to show the files on my hard drive, you can now see that I have 6 files saved on my hard drive. And there's a test folder there, which I made when I was testing this out. So this is an older folder which contains the files in case anything went wrong with downloading them or something like that. But I've tried it a couple of times now. It always works. But if you do a dir, so dir shows you the content of the current directory that you're looking at. So this one is just the 6 files that we downloaded. which is really awesome. So everything worked well. I hope for the guys that are following along in chat that everything went well, and that you guys don't get any issues with NCBI, because there's like 40 people actually watching the stream. Whoa, welcome everyone. So yeah, 40 people downloading it at the same time might cause a little bit of issues with NCBI. So just if it doesn't work now, try again in like 2 to 3 minutes, because it might be that we're overloading it. So just to make sure that that doesn't happen. All right, so you can use this for any data set on Geo. So if you look at another data set in Geo, it's always going to be the same. It's always going to be the same URL. The accession is just the ID of the sample, and then you just have to tell it which file you want to download, because you can download in multiple formats, but we're downloading in cell format, because that's the default format for Affymetrix arrays. All right, so I'm then going to use the affy package, right? So this is all affy. So the affy package has a function to read in cell files, but the only thing that we have to do, read affy, just reads all of the cell files in the current directory. So you don't have to give it a list of files that you want to use, but you have to add the phenotypic data. So the phenotypic data in this case is our samples array, right? Because we need to tell the function that we are loading in this data, and these are the control samples. These are measured at 0 time 0, and these are measured at time is 4 hours. So we need to create something which is called an annotated data frame, and we can use just the samples data frame that we just created. That's why I made it into a data frame, so that this conversion automatically works. So it's just a single command saying read all of the Affymetrix files in the current folder, and the annotation for it is the samples matrix that we just created. And then I'm going to store this in a new variable called cells. And then the next step is to normalize it. So again, microarrays, it's a very, very well established pipelines. So I can just say, well, use RMA, for example. There's like 6 or 7 methods that the package provides. RMA, I think, is the one that was used in the paper. So I thought, let's do what they do in the paper. So I'm just going to call the RMA function, and this function is going to call preprocess core. So this is where you would get the bug if you're under Linux. So if this call is giving you an error saying that pthreads error 22, then you need to go back and you need to install the preprocess core library. All right, let's go to R. Let's do this. So I'm going to read the cell files and then going to do the robust averages. So this should be really, really quick, right? They're very, very small files. So it will give you a warning message. That's fine. So it says, well, I'm doing background correction. So that means that it will look at all of the microarrays and look at the background intensity relative to the foreground intensity of the probes. It will do normalization. It will calculate all of the expression values for you. So if we just look at ESET, right, then we can see that it tells us this is an expression set. It has 8,793 features. So those are 8,793 probes on the microarray. There are 6 samples in total. The element names are expressions. So you can use XPRS on the ESET. And let's just show the first 10 to get the expression values. So here we see that this is the name of the probe. It is expressed at 8.7 in this one, and it is expressed at 8.79 in this one, and it's expressed slightly lower, 7.9 in one of the other samples. Right, so very basically, if we wanted to look at this, for example, using a little box plot, we could just make a box plot on the expressions that we found in our expression set. And you can see that it looks really, really nice because we've done the normalization. So RMA is really good at doing that. So and this is the point where you would normally do quality control on your samples to see if everything went okay. So some of the lectures I did before, we actually do this by hand. So we call preprocess core ourselves. We do our own log2 normalization. So you don't have to use the RMA function. You could do it yourself as well, but for the sake of not talking too much about microarray analysis, I thought it would be good to just use the package to get it over with, so to speak. All right, so now we have our expression sets. So now we just have to do the statistics, right? So the statistics, again, we're going to use all built-in default settings. So we're just going to say, I'm going to make a model matrix. And the only thing that I want to look at is the time. So the time comes from the P data, right? So that's the pheno data that we provided to the affy package. So what it will do is very basically, it will just look at every, it will look at the column called time, and it will say, oh, I see 2 different time points. So I'm gonna compare one time point with the other time point. Of course, if you have multiple, if you not only have control samples, but you have like P53 knockout samples or other types of samples, then this here you will make more complex. So you would say, I want to look at time, but I first want to correct for the sample type, or I want to look not at time, but I want to look at the differences between the different sample types that we have. We use limma to do differential expression, so I'm just going to say lmfit on the expression set using the design matrix that I just defined, and then I'm going to use eBayes to do the p-value computation, and then I have something which is called fit. You could call it anything that you want, but I call it fit because it's the linear model fit of the model to the data. And then I want to get all of the p-values. So for every probe on the array, I want to get a p-value back. So I'm just saying, give me the top table. So normally top table, if you don't call it with numbers infinite, it will just give you the top 10. You could get number is 15, then it gives you the 15 most differentially expressed ones. Why did you not build a contrast in this case? It does that. It builds a contrast. This is where it builds the contrast. Let me show you in R. So let me just go to R. So here, when I look at the design matrix, then the design matrix will say, hey, I have a contrast, and the contrast is time. So it has an intercept. So that's the average of a probe across all of the arrays, which it will drop when it fits it. And then it says time 4. So 3 time points were not time 4. 3 samples were at time point 4. Right, so it has a basic contrast test in there where it contrasts 4 hours versus 0 hours. And that is because it's a very, very simple data set. There's only one factor which is different, which is the time factor. So that's the only factor that it can do the differentially expressed genes on. So the top table, like I said, it will give us the top genes, but we don't want the top, we want all of them. So I'm just going to say number is infinite, and then it will give us back all of the data. So it will just give us all of the DEGs. So if I would look at the DEGs and I would just look at the first 10 of them, which is of course the first most differentially expressed, it will give us back a table which looks like this. So for each probe on the array, it will tell us this is the fold change. So this means that it's upregulated relative at 4 relative to 0, right? So 0 hours is our control and our base group, while the 4 is the other. So it's the other group, the upregulated group. The average expression is 5.9 of the probe. The t-test is this, the p-value is this, and then it adjusts it for multiple testing, and you also get a beta. That's kind of how it works. So there's the one group is 5.9, the other group is around 10, based on this. And of course, we can look at this probe. If we look back into our E set, into our expression set, then we can actually look at this probe. So you see here that it's very lowly expressed in the at 0 hours, and you see that it's highly expressed at the other one, right? The average expression over the array is 5.9, and the difference between these two groups is it should be 5.9 as well in that order. So we're just using the standard for that. So nothing fancy. This is how you would normally do a microarray experiment where you just have two groups. There's nothing really that you can do wrong when you only have two groups, because the model matrix is just simple. You could even flip the ones and the zeros and ones with twos and threes, and still it would understand what you're trying to do. All right, so now we have our differentially expressed gene. So we want to visualize this differential expression in a way. And the standard way of doing this is using a standard volcano plot. So in R, this is the minimum code that I could come up with to make a little volcano plot for this microarray. So what we are going to do is we're just going to use the plot function. We're going to say on the x-axis, I want to have the log-fold change. On the y-axis, I'm going to take the minus log 10 of the adjusted p-values so that I don't have to deal with multiple testing. I'm just going to take the multiple testing corrected p-values. I'm going to add my own label to the x-axis. I'm going to add my label to the y-axis. I'm going to give it a little main saying volcano plot. It's not a good name, could have been better, right? We could have said volcano plot 4 hours versus time point 0. And then I'm going to say, well, I want to use closed circles. So PCH is 19, which is the closed circle. The default is the open circle. And I'm going to say y-axis is I. That means that it won't give a little bit of extra space on the y-axis. So you can see that this point is slightly cut off or cut in half, different from the x-axis where you have a little bit of space towards the first point. But I don't like this axis here to begin a little bit negative, right? Because it starts at 0, half, 1. You can just cut it at 0. And then we add 2 lines for the significant threshold. So since we are looking at already multiple testing corrected data, we can just add 2 lines, one for the 5% threshold, one for the 1% threshold. So the one is green, 5%, and this is the blue one, this is the 1% threshold line. So all of these genes here are significantly upregulated at 4 hours relative to the start of the experiment. And the chances of having a false positive in this one is less, or is 1%. If we take all of these genes above 5%, we see that there are 4 genes which are downregulated relative to time point 0, and we see that there's 1, 2, 3, 4, 5, 6, around 10, 20 of them which are upregulated. I can't count exactly the numbers, but just a little bit of a visualization. Let's go to R. So the box plot is nice, but we can do the little visualization as well. And then it shows you indeed what we already saw before, that there are some genes which are upregulated, or actually quite a lot of genes which are upregulated, and there are some genes which are downregulated. So those are not the most interesting ones, because generally we want to see upregulation in response to the I already forgot the treatment, the neocarzinostatin, I would say. Anyway. All right, so little visualization, but we have our list of differentially expressed genes. So now we can start doing some annotation on them. So for that, we need to get the microarray description file, right? Because at the moment, we just have this, right? So we just have a name of a probe, but we have no idea which gene in the genome is being targeted. So we're going to automatically download this as well. If we go and look in Firefox, how do I know that I need to use GPL 201? Well, that's actually what it tells you here. It tells you here GPL 201, right? So it tells you which platform. So every microarray is slightly different. Every microarray has its own annotation file that is used to do the annotation of the platform. So if you would hover over the link, then it will tell you that this is the link to download it. So I just took this whole long address, and then I'm going to just save it into something which is called GPL 201-30390.txt, because this was the file name that I got when I downloaded it by hand. We then want to load it into R. So it's just a basic read table. It's just a tabular matrix. So we're just going to say read this table. There is a header in there. It is separated by tab characters. The first 16 lines are not usable. Well, they are metadata on when the file was created, who created it, which columns there are, but we don't want that for R, so we're just going to skip the first 16 lines in the file. It has quotes in there, so we have to disable quoting. And this is important because a lot of genes are things like 5' something, right? So there's a lot of quotes in the file which are not the beginning and the end of an entry in the file, but which are part of the name of a gene. So a lot of genes, or not a lot of genes, but some genes have like a 5' name. So or a 3'. So they have a quote in their name. So this is important. Make sure that you disable quoting, so that you don't allow quotes to be processed by R. And of course the row names is equal to 1, and that is because the first column in the file contains the name of the probes. So we want to use the name of the probes as the row names for this matrix that we're loading in. All right, so I think I need to download this file as well. This file is slightly bigger. I think it's 16 MBs, something like that. So let's start the download now so that we have that done. Should be relatively quick because I'm at home. It's actually really, really fast. But it could take a little bit of a time because this is a big microarray, or the microarray is relatively small, but the annotation is relatively big. All right, so now we have GPL201. So GPL201 is very basically, it's a big matrix, right? There's a lot of stuff in there. So we can just scroll. So you have a gene title column, which has the name of the gene written out. We have a gene symbol column where there's the gene symbol that we generally use. Then there's all kinds of additional annotations telling us, for example, where the sequence on the microarray came from, if it's a proprietary database or if it came from GenBank, which species are targeted, when the last time the annotation was updated. So since this is a relatively old microarray, the annotation is from 2014. So it's not very up to date relative to current standards, but when they wrote the paper in 2015, they probably used this annotation as well. So it's very, very good that we can use the old annotation for annotating the microarray to be able to exactly reproduce their analysis. And then there's a there's a GB accession. You can just ignore that. That's a different code. It's the GenBank accession for the probe if you want to go to GenBank and download the probe sequence. But you don't have to do that. All right, so that's kind of the layout of the annotation file. If we scroll all the way down, we can already see that there is gene ontology in here, right? So this is a gene ontology code. So that's what we're going to use later on to make sure that we can annotate every probe as being part of a certain annotation, right? So we have now defined our set. So the set of genes is based on differential expression, and the annotation is going to come from this file to use in the overrepresentation analysis. All right. So of course we want to make sure that we get a little table of differentially expressed genes so that it looks nice and we can, for example, put it in a paper. So what I did is I just take, I do cbind. So I say add an additional column to the table that I already had. So I'm going to add a symbol table. And since the differentially expressed genes, so the matrix that we made here is annotated using the probe names, we can very basically just say, take the row names of these and then take the annotation. So resort the annotation so that the file that we have matches the row names of the differentially expressed genes matrix. And in this case, I just want to have one column from the annotation added. So I'm going to say take the column called gene symbol out of this big matrix and then combine that with the table DEGs and just take the 3 columns that we're interested in. Right? I'm only interested in the log-fold change, the p-value, and the adjusted p-value. Well, in theory, you could drop the p-value as well because I always use the adjusted p-value. But at least now we have a little table, and this table could go in your supplemental files of your paper, or this table could be one of your main results tables in the paper when you show, for example, the first 10. All right, let's do this. So I'm going to go back to R. I'm going to say add this little table. Right, so I'm just going to take the annotation, make sure that we are using the same ordering. So in this case, the most differentially expressed genes is called CXCL8. It shows a log-fold change of 3.6, and it has a p-value adjusted of 0.0004849, depending on how you round it. So that's really good. And of course, we can look at how many genes are actually differentially expressed. So under 1% adjusted p-value, we find that 7 genes are differentially expressed. So a very small amount of genes. And this is, of course, based on the fact that we only have 3 samples versus 3 samples. Right? If we would have like 30 samples versus 30 samples, we would probably be in a much better position to assess differential expression in the genes. But since we have only 3 versus 3, this is kind of the maximum that we can do at this point. All right. So a little intermezzo, because now we are starting to deal with the enrichment analysis. So I have to tell you guys about gene ontology. If you want to know much more about gene ontology than what we're doing today, I actually did a lecture about gene ontology. So it's this video. I think I put it in the description. So if you're interested in learning all of the ins and outs of gene ontology and how it's used and different pathways like KEGG, then watch the video. It goes into much more detail. But very basically, here we see a gene ontology kind of annotation, right? So gene ontology is a tree structure. So you have a top level term, which then gets subdivided into smaller terms. And these smaller terms can then be connected to other terms in the tree. So there's a relationship between each of these. So it's like, so a metabolic process is either a small molecule metabolic process, a primary metabolic process, an organic substance metabolic process, or a biosynthetic process. So those are the metabolic processes that there are. And then, for example, the monosaccharide metabolic process is an example of a small molecule metabolic process, but it is also a carbohydrate metabolic process, which in turn is part of the primary metabolic processes as well as the organic substrate processes, right? So all of these terms are connected to each other in a certain way, and this is how this tree structure is functioned. So you can just read the tree, so from the bottom to the top. So for example, if I want to know what a hexose biosynthetic process is, then it is part of the hexose metabolic process, but also the monosaccharide biosynthetic process. Besides that, we have 3 root annotations. So you can do gene ontology based on cellular component. So this is the localization. So where is it located in the cell? So it could be the nucleus, the cytosol, endoplasmatic reticulum, cell wall. It can be an excreted protein. It could be a protein in the mitochondria. Then we have the molecular function. So the molecular function is a group, is an ontology which defines the function of a certain gene at a molecular level. So the molecular function could, for example, be DNA repair, or it could be protein degradation, right? So that is a metabolic molecular function. So a molecular function is just a function that this group of genes does together. And then we have biological process. So biological processes are more like pathways. So they are groups of genes that work together in a certain biological process. So a biological process is, for example, metabolism, or it is movement, or it is reproduction, right? So those are very high level terms, which are then get subdivided into smaller and smaller terms. So like I said, if you want to know more about gene ontology, you can watch the YouTube video where there's much more detail about how it exactly works. So we already saw the data in the GPL file, right? So in the array annotation file, you have gene ontology annotation, and each of these lines is structured like this. So in the column for a single probe, you have the ID of the gene ontology process, then you have the name of the process, and then you have the source. So where the how this gene got to be in this group, right? So you have, for example, well, this probe here, don't know which one it is because it's not in the table, but it belongs to gene ontology group 00015558. This group is the regulation of cell growth group, and it is inferred from electronic annotation. So it has not been validated, right? So it's just an electronic annotation. So some AI, or well, probably not AI, but some process, electronic process, annotated this gene as being part of this group, which is different from the second one, right? So the second one is a probe which has been annotated to the mitotic cell cycle, but this has been done by author statement. This means that this annotation comes from a paper that has been published, right? So this is a stronger annotation than the inferred from electronic annotation. So some gene ontologies are very certain, some of them are less certain because they are done automatically. But if we look at it, we can see that every one of these are separated by 3 forward slashes, right? So we have an ID, a name, and then a source, 3 forward slashes, and then an ID, a name, and a source, right? Because it is a tree, right? So you have to, not a single probe can belong to many different gene ontology groups. So how do we deal with this, right? Because we just have our table. So we need to write a little function which parses out this gene ontology data for a certain probe. So if we would want to do this by hand, then I would say, well, if I want to know for a certain probe what gene ontologies there were, I want to first split on the 3 forward slashes, right? And then I get like 10 things back. So if this thing would have 10 different GO annotations, when I would split on the forward slashes, I would get 10 annotations back. And then, of course, if I want to get the individual parts, then I can split on the 2 forward slashes to then get the ID, the name, and the source. So I wrote a little function to do this automatically based on how Affymetrix puts this annotation in their files. So that is what we're going to do. And why are we going to do this? Well, this is because R does not like trees. If you're programming in R is very good with vectors and matrices, but R kind of sucks when it comes to tree structures. So it is better in R to just have a massive matrix where you have a probe, an annotation, and a description. And then we have the same probe, another annotation, and another description. And then we have the second probe, which has its own GO number and its own description, right? So we're going to take this data, which is in this format, and then for each probe, we're going to make a little matrix. So we're just going to repeat the probe names. So for example, if we have 2 GO annotations for the first probe will be in the list twice. If it has 10 GO IDs, it's going to be in the list 10 times. If it has 10 descriptors, it's going to be in there 10 times, right? So R really doesn't like trees. So we're going to have to avoid this. We're going to go from a tree structure into kind of a 2-dimensional matrix structure. So a basic matrix is much better for R. All right, so don't be scared by the code on the next slide. This is the function that we're going to use. And it is very logical. But when you see it for the first time, it can be really overwhelming. So it is just a function that I define. So I put the whole function into something which is called getGeo, right? So it's a helper function. We will take a single probe and we need to specify which gene ontology we want. So if you look at the table, the GPL 201 table, it has 3 columns. One for biological process, one for molecular function, and one for cellular localization, right? So it has 3 columns. So in this case, we're going to just do 1 column. So we're just going to say, take the biological process gene ontology for this probe that I specify. So like I said, the first thing that I'm going to do is I'm going to split on 3 forward slashes. So since I'm splitting on 3 forward slashes, so I'm just going to use the string split function. So I'm going to say string split from the GPL 201 matrix, take this probe, take this ontology, and then string split it by 3 forward slashes, and then take the first element that comes out. So the first element that comes out, and this is important, is not the first ID1, name1, source1. No, it's the whole split, because string split assumes that you are going to give it multiple lines of text. But we're going to give it only 1 line of text, and that is why we have to have these double bracket 1, close double bracket. We could have also just put unlist here. Then it would make a list of length of 1 into a list or into a vector which has the same list. But I'm just going to say string split this probe, this ontology, using the 3 forward slashes. And then I'm going to store it in a variable called split. And then I'm going to use a little trick. So what am I going to do? Well, I want to parse out the GeoIDs and the descriptions. So I'm going to say GOS is going to be my GeoIDs, and the DES is going to be my descriptions. So again, we read from the inside out. So I'm going to take my split and then split it again based on 2 forward slashes. And then I'm going to select from that. So I'm going to lapply to the thing that comes out a selection, and I'm going to take the first element, right? So the name is the first element in the list. And then I'm going to unlist this whole thing, and I'm going to do trim WS. So trim WS removes all of the white spaces at the beginning and at the end. And this is because sometimes there's a space at the beginning and there are spaces in between here as well, right? So you can see that it's source 1, space, 3 forward slashes, space, ID2. So ID2, when I split it, it comes as space, ID2, space. And I don't want the spaces at the beginning. I don't want the spaces at the end. So that is why I can use the trim WS. And then this will give me the GeoIDs. The same thing for the descriptions, but now instead of selecting the first element, I'm going to select the second element. If I wanted to get the annotation, right, so how it was annotated, so the source, then I would repeat this line again, and then I would select the third element of the list. And then for each of the geos, I would know geo 1, description 1, source 1. But I don't want to know the source because we're not going to deal with the source. We're going to assume that every annotation is perfect and that there's no kind of weight that we have to put on there. All right, so then I'm going to make an if statement because I have to make sure that this probe actually has a geo annotation. Not all probes have geo annotations. So, but if that's the case, so I'm just going to check the length of the GeoIDs that I get back. So if the length of the GeoIDs is larger than 0, it means that this probe had geo annotations available. And then I'm going to just make my little matrix. So I'm now going to say, well, cbind the GeoIDs with the term geo double point because they're just numbers. So I want to say geo double point and then the number, and then add a secondary column which is called description. And then I'm going to take the name of the probe and then repeat it for every row of the matrix that I just created, and then add that as the row names. And then I'm going to return this little matrix. If there is no gene ontology annotation, I'm just going to return nothing. So I'm just going to say return nothing, right? So only when the length is larger than 1 am I returning a matrix? Otherwise, I'm not returning anything. So it's a complex function, but it works for all of the affymetrics arrays that I've seen so far. It's relatively straightforward to do, and I hope that it's understandable after me explaining it to you guys. I hope that you kind of see the rationale on seeing this structured string within the column and then splitting it. And the way that I generally do this, right, if I would go to R, I would say GPL 201, and I want to have this column. So let me make sure that I check out the correct column. So it's called Gene Ontology Biological Process. Right, so it's like this. And let's just look at the first one. Right, so if I look at the first one, then here you see for the first probe all of the annotation that is there. Right, so generally I just build it up one by one. So I say, okay, so I see that the main thing is, so the main split is these 3 forward slashes. Oh, slightly different keyboard. So 3 forward slashes, so I'm just going to string split it, right? And then I see, okay, so now I get a list of everything split it out. So I have a list of 1 because I only did 1, I didn't do 2. So it has 23 annotations. So I'm just going to remove the list from it. So now I have 23 annotations. And then I'm going to do, so I just build it up one by one, right? So that's generally how I code, just so that you guys are aware of how you could do this yourself in case you encounter a different type of encoding, right? Because this works for affymetrics arrays, but in case you wanted to use other arrays, the annotation might be different. All right, so next we're going to build a matrix for all the probes, right? Because this function just does it for a single probe, but we have many probes on our array. We have like 8,000 plus probes. So I'm going to build a little for loop. So I'm going to say, well, I have a GO matrix, which initially is just empty, so just an empty matrix. And then I'm going to say for each probe in the row names of the differentially expressed genes, I'm going to say row bind to the matrix that I had, the getGO of this probe, right? And I'm not specifying the second parameter because the second parameter defaults to being the biological process. And in this case, that's good enough for me to look at the biological process. And then I'm just going to say, well, after this loop is done, look at the first 10, and then this is how it looks, because we already knew that the first probe on the array has 23 annotations. So it's all the same probe, all with different GO annotations, and then with the different descriptions of these GO annotations. So this will take a little while. So let's go to R, and let's start building this matrix. Because it will take some time for this for loop to run, because it's relatively inefficient, right? Because you can imagine that I'm now building a matrix which is around 8,500 times, well, 20-ish GO terms per gene. So it's going to be 20,000 rows, something like that. It's going to be a big matrix. So it's going to take a little bit of processing power to do this. But it's not too bad. If you want to do this for an array which has 100,000 probes, you would want to do it slightly differently. You don't want to use the naive approach of building a massive matrix at that point. You probably want to pre-allocate the matrix. So in the end, like took a minute or something like that. Not too bad, not too bad. All right, so that's it. So we have our matrix. So this matrix looks like this. We could look at it in R, but this is just the screenshot that I took. So you just have to trust me that it will look like this. All right, so now back to some theory, right? Because we're now ready to start doing our enrichment analysis. So enrichment analysis is based on this figure here. So this figure here shows everything that you can know about enrichment analysis, right? So for each GO term in our list of differentially expressed genes, we can define the following, right? So I just define a set saying I have, so in our case it's around 20 genes, 30 genes which are differentially expressed. And I now want to compare if any of the annotations within this group of 30 is different from the whole microarray, right? So first is U. So U is the total number of probes. So that's the first circle. So the first circle is just the number of probes on the array. So in our case, the number of probes is, for example, 8,500. Right? And then we define something which is M, and that is the total number of probes with the GO term that we're currently looking at, right? Because I'm doing this per GO term, because I want to know for the first GO term, is this one overrepresented, yes or no, right? So M is all of the probes which also have this GO term. K is the number of probes in our list, right? So this circle is part of U because it belongs to probes on the array, right? U is total probes on the array, and K is the circle, so it's the number of differentially expressed probes that we have. So in our case, we might have 20 or 30 or 40 differentially expressed probes. And then we look at X, right? Because X is the overlap between K and M. So it is the number of probes that have a certain GO annotation, which are also in our list, right? So it's so for example, X divided by K is the ratio of probes that have a certain gene ontology, right? So it's like 5 out of 20 differentially expressed genes have this GO term. And then we also define a second parameter or an additional N. So N is defined as big N minus M. So it is the number of probes which is not in our list, right? Because M, it is the number of probes not in our list. So then for each GO term in our list, we can additionally define the foreground ratio. So the foreground ratio is X divided by K. So it's the number so for a single GO term, it's the number of genes which have this GO term divided by the total number of differentially expressed genes. And we can also compute the background ratio, which is M, right? So all of the probes which have this GO term divided by all of the probes on the array, right? So now the question is the foreground ratio significantly more than the background ratio? That is our whole question that we want to have answered. So for that, we can use a hypergeometric test. So I'm very sorry for showing you guys mathematics on a slide. Generally, I try and avoid showing you guys mathematics, but in this case, it is a really nice analysis. So this is from ZAR JH from BioAnalysis 1999, and it is actually quite well written. So it is very understandable to know how to do a hypergeometric test, right? So again, we have our figure here with all of the probes with a certain GO term, all of the probes which are differentially expressed, and then all of the probes which are differentially expressed and also have our GO term, right? So now the question is, if I select K probes at random from U, what is the probability of obtaining X? So this is wrong. This needs to be X. I'm very, very sorry. Slides, sometimes K and X, you turn them around. So when you select K probes at random from U, the probability of obtaining X is K divided by X multiplied with U minus K divided by M minus X. That is just a kind of standard binomial distribution. So now the probability of finding more than X probes in a GO term is just the same formula, but now we just add up all of the probabilities from I equals X to I equals the minimum of K and M. So this is how you would compute it, right? So it's just very basically this formula. You fill in I equals x, then you fill in I equals x plus 1, then you fill in I equals x plus 2, right? So it's very basic. You're just adding up the probability of finding 7 out of 20 to be in your GO term, the probability of finding 8 out of 20 of being in your GO term, plus 9 out of 20, and so on until you reach 20 out of 20, right? So you're just basically dividing it and saying, well, there's a probability that of finding x, and then all of the stuff which is above x. So we don't have to do this at all, right? We don't have to write this from scratch. So we can just use the p-hyper function, because this will give us the probability of this hypergeometric test. So there's only a little bit of an issue, because R computes the opposite. So R does not compute the chance of finding x or more, it computes the probability of finding x or less probes in a GO term, right? So you would normally want to say, okay, so it does an underrepresentation test, right? And that's not what we want. We want to know what is the likelihood of finding x or more gene ontologies relative to the background ratio, right? Fortunately, the p-hyper function also has an option to flip it around, right? So you can just take the thing, flip it around, and that is how you can do it in R. All right, so let's start coding again and make sure that we code this in from scratch. So first, we can only use probes that have gene ontology, right? So we have to make sure that we just define this GO matrix, that we only use differentially expressed probes, differentially expressed genes, which are also found in the row names of the GOs, right? So I'm gonna say, take my differentially expressed gene list and then only retain probes which are also found in the GO list. Because probes which do not have a GO annotation, they don't add, right? They're extra, so we can just get rid of them. So it means that we don't really have information on them. So if we have no information on them, we can include them in the test that we're going to do. And then we need to define our foreground and our background, right? So the foreground is just saying, well, I'm going to take all of the differentially expressed genes for which the adjusted p-value is lower than 0.05, and then give me the row names of those. And then the same thing for the background set, but the background set is going to be all of the genes in the differentially expressed gene list, right? So since we asked for the differentially expressed genes sorted from kind of highest or most differentially expressed all the way to stuff that is not differentially expressed. And so when we did the top list, we actually said, give me infinite back, so give me everything back. And then what am I going to do? Well, I'm going to just say, well, I now have all of the probe names which are differentially expressed in my FG variable, and in my BG variable, I have all of the probe names which are in the list, right? So in the total list of genes. And then I'm just going to say, well, now I can take my GO matrix, use the probes from the foreground, and then get the GO IDs and put them in fg.go. And I'm going to do the same for the background. So I'm going to take from the background, I'm going to take the GO from the GO matrix. I'm going to take the background set of probes and then ask for the GO identifier and then store that in background.go. All right, let's switch to R. Let's start doing this. So we built our GOs, right? So just let's check this, make sure that everything went okay. So 1 to 10. So indeed, we can see here, these are the first 10 annotations. These are the GOs. All right, so make sure that we throw away. So let me show you. So DEG. So DEGS. So initially we have 8,793 probes. If we now throw away all of the probes which do not have a GO annotation, we end up with 8,328. So that means that there's around 470 probes which do not map to a gene which have a geo annotation. So we're just going to leave those out of the analysis. All right, then we're going to define our foreground and our background sets. And now we can ask questions like foreground.geo, just show me the first 10. Right, so here we see that, well, for this probe, these are the geo terms, and we can see that for each of these, we have our geo terms. Right, so for every probe, we have all of the geo terms. So we have just a list of that. Same for the background. All right, so let's do our test. So again, picture on the top, because we can then remember what U is, what M is, what X is, and what K is. So now we can just define these per gene ontology. So that's what I'm going to say. So for each gene ontology, which is found in the foreground set, so I'm going to say, give me the unique values in the foreground set geo. So this just gives me a list without any duplicates, so that I go through each geo term just once. Right? So I'm going to say for a geo term in all of the geo terms which are found in the foreground, what do I want to do? Well, I want to define X, right? So I want to define X here. So this is, I'm going to ask which fg.geo is equal to the geo term, right? So I'm just going to ask how many of my foreground geos are similar to the geo term that I'm having. And then I'm going to use this, ask for the names of the FGO, right? Because the names are the probe names. So I'm going to go from geo to probe name. So from geo identifier back to probe name. And then I'm going to ask unique. So I'm just going to ask how many probes in my foreground set have this geo annotation. And then I'm going to ask for the length, and that's going to count them. I'm going to do the exact same thing for the background. So I'm going to say which background geo is geo, and then use that to select from the names of the background geo. So this will give me M. It will give me all of the probes which have this geo term. I'm going to compute my N, so small letter n, right, which is U minus M. So this is U, the length of all of the probes in the background set, right? So I'm going to say names of the background geo, unique, the length, and then subtract M from it to compute N, which is everything outside of M. And then I'm going to compute K. So K is going to be all of the probes which are in the foreground set. And then I can do my p hypertest. So the way that I flip the p hypertest around is just saying lower tail is equal to false. But from X we have to subtract 1. Right? So I just give p hyper X, M, N, and K. And then I tell it lower tail is equal to false to flip the test around. But the test is asking me, is X smaller than or equal to the number that I observe? So the smaller than or equal, since we want to know larger than or equal, we need to subtract 1. So now it gives me smaller than or equal to X minus 1. And then we flip it around, which means larger than X or equal to X. So this is going to just give me a p-value, and I'm just going to then add everything into a massive matrix. So what am I going to say? Well, I'm going to remember X, I'm going to remember M, I'm going to remember U, so N plus M, I'm going to remember K, and I'm going to remember P. Right? So I'm going to, all of these values, I'm going to combine them into a vector, and then I'm going to row bind this vector to the ORA matrix that I'm defining. Right? So it will go and it will do the first geo term, it will add 1 row to the matrix. It will do the next geo term, will add another row to the matrix. So then it will take the row names of the overrepresentation analysis. So the row names of the overrepresentation analysis, I'm going to set to the unique FG foreground geo terms, right? Because that's what I was iterating through. And then I'm going to add column names to remember what's in each column. So I'm going to say X, M, N plus M, K, and P, right? Because those were the values that I computed. So those are the values here. I could have just called this one U, but I like N plus M. And why did I not use M plus N? It's because otherwise I might get confused and select the wrong column. But N plus M seems to be okay. All right, so let's go to R. Let's do this and just run through the pathway overrepresentation analysis, right? All right, so this is actually really, really quick, right? This is like, it does it, and why is that? There are not too many GO terms in the foreground set. So it doesn't have to do it for all possible GOs, it just does it for the foreground set, right? So that's the nice thing about this. So then if we look at ORA, right, it looks like a matrix like this. So there's still like a lot of terms, right? So we actually see here that GO, there was one that was in X, M is actually 126. The universe is always going to be the same, 8,328. So those are the number of probes with gene ontology annotation. And in this case, K, so thinking back about the circle, so K was 60. So I find 1 out of 60, and in the background I found, so in the foreground I find 1 divided by K, so 1 out of 60, and in the background I find 126 out of 8,300. And this is not significant, right? So the chances of pulling 1 out of 60 relative to having 126 out of 8,328, this is a non-significant difference. So it will tell me that for each of the GO terms. So if we scroll, we already see that some of them indeed are kind of significant, right? So 2 out of 19 is 2 out of 60 when there's only 19 out of 8. That is a significant. But we only have the GO terms here, right? So we need to make sure that we annotate this and build a real matrix which we can use. So the first thing that I'm going to do is I'm going to compute an adjusted p-value, right? So I'm going to say adjust the values in the p column using Benjamini-Hochberg and then add a new column to that, right? Because I want to make sure that the values that I'm looking at are multiple testing corrected, right? Just like all of the other values for differential gene expression. All right, so I'm going to bind to the aura column a new column, which is the adjusted p-value. And then I'm going to say, well, I only want to know the ones which are significant. So I'm going to say, well, when the adjusted p-value is lower than 0.05, at that point, call them significant, right? So only give me the row names which are significant when I adjust for multiple testing. All right, and then I'm going to take the annotation of the significant GO terms. So I'm just going to look up each of the GO terms in the GOS matrix, right? Because the GOS matrix just has probe name, GO term, description of the GO term. And of course, every GO term will have the same description. So I'm just going to go for each of the significant values, and I'm going to add a name. So I'm going to say, well, take the, so I'm going to ask which GO is I, and then I'm just going to take the description column, and I'm going to collapse this down to a single description instead of 5 or 10 descriptions, which might be there. And then I'm just going to add them to it. So, and then I'm going to add the description to my ORA table, so to my overrepresentation analysis table. All right, so we're almost there. So let me go back to R and add the descriptions, right? So now when we look at the ORA table, so now it has the GO term and it has the description, and it also is now reduced to only the ones which are significant after multiple testing correction. All right, very, very good. So let's format the output table, because generally people think in terms of observed and expected values, right? So we have X1. So X1 is, of course, the interesting probes with the GO term divided by the number of interesting probes, so the number of probes in our set. And then here we have the background ratio. So this is the foreground ratios, the background ratio. So all of the terms, or all of the probes that have this term, divided by the universe. And then we compute our expectation. So we can actually take x2, which is the observation in the background, the ratio, multiply this by the set or by the size of our input set. And this is, of course, how many we expect, right? If you have 3 out of 10, if I have 5 draws, then I would expect 1.5. If I have 1 out of 20, if I would draw 10, then I would expect half of them, right? So it's a ratio. So if you have the ratio in the background set, you can multiply this by the size of the foreground set to get your expectation. And then we're going to put it all together into a single table. So I'm just going to say, well, the ORA table is going to be the description, the expected value, the observed value, the background value, the fold change, which is the ratio of the ratios. And then I'm going to add the p-value and the adjusted p-value and then make it into the data frame. And then it looks really, really nice because then it just looks like a real output table, like something that you would get from David, or you would get from WebGestalt or some other tool that will do gene ontology analysis for you. All right, so let's do this in R to make sure that we have the nice output table so that you guys can see that as well. So this is then how it looks like. And of course, this table, we can just use write table, write it to the hard drive, and then load it into Excel or something like that. So that's it. That's how hard it is or how easy it is to do gene ontology pathway over representation yourself. So I had some closing remarks here. So some observations about the output that we get. So one of the things that we observe is that we have a relatively small foreground set, right? Our foreground set is around 60 differentially expressed genes. So we could think about some more relaxed thresholds, right? So instead of using 0.05 for defining a gene as being differentially expressed, we could actually say, no, we're gonna take 0.1, right? So that will increase the size of our foreground set a little bit. And of course, if we would have more samples, we would have a more accurate differentially expression list. And that would give us a better foreground set, right? We have only 3 samples versus 3 samples. So in the end, it's very, well, it's not shaky, but it's just more samples will give us more statistical power, which means that we have more certainty in the output. That being said, we already see some interesting geoterms, right? We know that we have a cancer cell line on which we throw an anti-cancer drug, right? So here we see negative regulation of transcription. That is something that we would expect, right? We would expect neocarzinostatin to kind of reduce the transcription of the genes, right? Or we would expect to have like things like cell death or something come up. One of the other things that we see all the way at the bottom, we see response to molecule of bacterial origin. And that is actually a very, very accurate term to come up because neocarzinostatin, the molecule comes from bacteria, right? So it makes sense that the response to a molecule of bacterial origin comes up. So it's not like the end all to all annotation problems in biology. However, we already start learning some things. So my advice to you guys is repeat the analysis also for the other 2 ontology terms, not just for the biological process, but also for molecular function and also for the cellular localization. If you do cellular localization as something that I found interesting, if you do cellular localization, it points directly to the nucleus, right? So it points you and says something is going on in the nucleus. Which is interesting because this drug is indeed a drug which works on reducing DNA replicating capability. So where to go next, right? you might want to do KEG, or pathway overrepresentation analysis, relative to gene expression, or to gene ontology. It is just a different annotation, right? So again, you would make a different GOS matrix. So you would replace the GOS matrix by something where you would have the probe mapping to a KEG ID, mapping to a KEG description, right? KEG has pathways which are called MMU or HSU for humans, but it also is just a number, right? So KEG has a whole set of pathways. Genes are either part of a pathway or they're not part of a pathway. So it's just an annotation, just basically like gene ontology is. And of course, if you want to improve this algorithm, then of course you could run it for upregulated genes and then run it for downregulated genes, right? Because we're now grouping all differentially expressed genes together. But it might be that a certain gene ontology term is only found in the upregulated gene, while a different gene ontology term is found in the downregulated genes. So splitting up and downregulated genes from each other might improve your ability to see things. We can also use the effect size or the p-value to give more weight, right? You would imagine saying that, well, if we look at genes which have a p-value under 1%, we're going to put in this twice. So we're going to put in the ORA terms twice for this one. If they're only below 0.5, we're just going to put it in once, right? So you could weigh, or you could add more categories, or you could duplicate the entry for certain genes, right? So that would make it bigger. And you could use the effect size for this as well, saying that everything which is having a fold change higher than 4, it gets 4 entries, while everything with a fold change higher than 2, it gets 2 entries. Everything with a fold change higher than 1, it gets only 1 entry, right? So this is a way to kind of make the algorithm incorporate the effect sizes that you can see in the test that it does. And then there's also called something which is iterative enrichment based on ranking, right? In which you would start with, for example, the top differentially expressed gene, do the analysis, then do the top 2, do the analysis, do the top 3, do the analysis, do the top 4, do the analysis. And then you would see across the whole set when a GO term comes up. So, because now we're just taking a single cutoff, looking at p-values for certain GO terms. But of course, you could start with look at the top 1, top 2, top 3, top 4, top 5, and so on. And every time run the analysis so that you get an idea of where the best cutoff is going to be for gene ontology to describe the data set that you have. And these last two, when you use the effect size / the p-value, you're going more into the direction of weighted co-expression network analysis, so WGCNA, weighted gene co-expression network analysis. And that also uses a combination of p-values to add more lottery tickets to the pool, and they also use kind of an iterative enrichment based on ranking to kind of have a better annotation, so to speak. All right, so 1 hour, 45 minutes. Not my longest stream, not my shortest stream, but yeah, this is kind of pathway overrepresentation analysis. So that's what I wanted to tell you guys. I like it. It's nice. It's fast. It's fun. It gives some biological meaning to just having a list of differentially expressed genes. So that's what I wanted to talk to you guys about. All right, let me look at chat. Thank you. Let me just pull it up on the main screen. Thank you for the content that you've put in this channel. Can I have your email? Yeah, so my email is just on the About page. So just send it to my university email address. So I'm located at Northumbria. So it's denny.arons@northumbria.ac.uk. But it's on the About page of my channel as well. So no worries. All right, what would be an approach of going about performing data integration of transcriptomic and metabolomics data? Yeah, that's a lot of people are looking at that. So I have my own views on that. I'm currently building an R package for that exact reason. So data integration is hard because transcriptomics and metabolomics data are not directly connected to each other, right? Like it's not like proteins, transcripts, and DNA, right? Because DNA codes for an RNA, which codes for a protein. But a metabolite is a molecule which is modified by proteins, or not even proteins, because it could be like short non-coding RNAs. So it is an unsolved question. I'm working on it, and I have some ideas on how to do it. There are some R packages out there which claim to do proper integration, but I'm currently writing my own. So drop me an email, and we can have a Zoom or something like that to talk about what you exactly want to do and how you wanna do that. Because I kind of like thinking about it, but it's an unsolved problem. So feel free to reach out and talk about it. Congratulations on your wedding and welcome back. Oh, thank you. Yes, I totally forgot. I also didn't put in a picture. So yeah, I had my wedding. I got married on in April. So yeah, not just busy with work, but also busy with life. So on another note, I went to a concert yesterday, so I'm still having like my ears ringing and my voice is probably still a little bit rough. But yeah, it's good to be back. It's good to be back, and I'm definitely gonna make more streams. And I would love to thank you guys. So this is the overview of today, and yeah, of course, if you like the stream, leave a like. It helps the discoverability, so other people can find it as well. And don't forget to subscribe to the YouTube channel for upcoming content. So thank you guys so much for bearing with me, and I will say see you next time. So ciao.